Okay. Whoa. Okay, so you're probably wondering who we are, and so hopefully future nanoscientists. Um, but in terms of names, I'm Amy.
I'm Janine.
I'm William.
And so on Wednesday, we studied how silver nanoparticles affect yeast cells and biotoxicity. And so biotoxicity is pretty self-explanatory. It's basically studying things that are toxic to various organisms. And silver actually has a very special ability to kill single-celled organisms, and it's in a lot more things than you think. But it's been present and used in the same way as antimicrobial throughout forever. So basically, I think it was the Romans, but a lot of people throughout history have put silver spoons in the bottom of milk to keep it from spoiling, or placed spoons with their meat to keep it fresh. And so it's basically just it kills the bacteria, and we'll get into more of how that works later. But it's also in teddy bears, which you might not expect. Manufacturers realize that, oh, this thing kills germs, so at least most germs. So let's put in the teddy bears so your kids won't get sick. So it's in a lot of things just to keep things cleaner and just generally safer. And on another note, it's actually used in beauty products sometimes, and it's in other antimicrobial creams. So there's a lot more specifics to this, so Janine will get into that.
Okay. This week, as basically everyone has said, we had to draw pictures for homework. And so this lovely picture right here is definitely check plus quality. And so Wednesday, we learned about biotoxicity of nanoparticles, and as Amy mentioned, so during our lab section, we tested different types of silver and how they damaged single-celled organisms, which in our case was yeast. So we tested silver powder, silver nanoparticles, and silver ions. And this analogy represents the results based on our different types and methods of entry into the yeast. So the theater on the top, far left, depicts the nanoparticles. Nanoparticles are transported through endocytosis, which is basically when a cell engulfs particles like food in a sort of capsule, like a bubble, and then it brings it into the cell. And so this is represented by the person in front on top hiding the other people within the large coat.
Top hat.
Yeah, the one with the top hat hiding the other people. And so then the person enters through the door, and then the nanoparticles will be released into the theater, which is the yeast cell. And so in the middle, on the top, those represent the ions, which move into the cell by ion channels. The ion channels usually transport small positive ions like sodium or potassium. And so this analogy shows this by the people going in through the front door. And an important feature to note of this drawing is that this is a different door than the one the nanoparticles used. And that's because they have different methods of entry, endocytosis versus ion channels. And so on the far right, this represents the silver powder. And so ions have to break off the larger silver particles in order to enter the cell. The silver particles are actually larger than the cell, I believe. And so in order for it to affect the cell, ions have to break off of it. And so this analogy shows this by the people going in, through the clump of people at the bottom, which is the powder. And then one by one, they're breaking off and going in through the door. And the method of entry is the same, the ion channel, because the ions are breaking off. So now, if we look at the bottom half, this shows the extent of the damage each type does, which we found out during the lab. And so the left side, nanoparticles do the most damage to the cell, which happens in real life because they are the smallest and fastest particles to enter. And so ions do the second most damage because they, nanoparticles also have the ions that break off, but that happens inside the cell. And so that happens faster because nanoparticles are a lot like smaller, they have a higher surface area to volume ratio, which makes them unstable. And so ions do the second most damage because they're slower than nanoparticles. They have to go through the ion channel. And then the one that does the least damage is the powder because it doesn't directly enter the cell. Ions have to break off of it and then enter the cell, so it's very slow. And so William will explain a little more about the experiment and then other key biotoxicity concepts.
Yeah, so a little more things to know in addition to this analogy. So first of all, we also tried the same experiment but with gold nanoparticles, and we found out that the gold nanoparticles actually don't do any damage to the yeast cells because the gold nanoparticles can't attack the yeast cells due to their they can't get oxidated as quickly as the silver nanoparticles. And I'll also explain how the silver nanoparticles penetrate the cell. So first, the silver nanoparticles enter, their ions enter through ion channels, and then the ions react with the oxygen inside the cell, and then the nanoparticles form reactive oxygen species that prevent the yeast cells from performing cellular respiration and also break down the cell membrane. And so the yeast cell dies due to both protein dysfunction and the breakdown of its cell membrane caused by its inability to perform cellular respiration. And I also wanted to explain the manometer, which we use to measure the amount of respiration the yeast cells had. So basically, you have like a U-shaped container, and we basically fill it with liquid, and on one end of it, we put the yeast container, and then we completely seal it with parafilm, and then we can measure the yeast cells' respiration by looking at the water level change on the left of the U. So that's how the manometer works. And do you two have anything to add to that?
Thank you for listening, everyone.
All right, thank you.
I think we're done.
That's like the most important thing you can say in a talk is the thank you for listening, because otherwise the audience is paralyzed with fear, not knowing whether it's time to clap or not. So you have to save the audience, and you have to eventually say, thank you, you're allowed to clap now. Okay, questions?
So this is a non-expert question. What are the practical uses of each of the different forms of silver, if you know?
Well, they are able to kill single-celled organisms, and in this experiment, we used yeast. So once we introduced the yeast, it was able to kill the organism. And so basically, if you add a layer of silver particles, silver nanoparticles, it's able to kill the single-celled, the microbes and the single-celled organisms there. So it basically it cleans it sort of, but just like in a different way.
So I know that you mentioned some things, but other than teddy bears, do you want to mention some practical products that have silver in them?
Yeah, there's the microbial cream that I mentioned, and it's you can get it at CVS, and basically it's used to help dress wounds and clean it so it doesn't get infected.
There's Band-Aids.
Yeah.
There's gym socks. Okay. Other questions? Okay. While you're going, I'm going to ask a question as I walk. So yeast is an example of what kind of organism? What are the other things you might bump into that is in the same family as a yeast?
prokaryotes, like
Well, I mean, so like there's bacteria, you say microbes. Yeast is not a bacteria.
Right.
It's an example of another sort of big class.
fungi.
Fungi, yeah.
Yeah.
And that's sort of the stuff that often, well, both the fungus and the bacteria stink,
Right.
but they're both part of the things that make like gym clothes stink. Okay, I was coming over here for somebody's question, but I don't remember who.
So given the human body is also made up of cells, is using silver going to kill me eventually?
So no, because we have a lot of cells. But actually, an interesting thing is that like those creams that Amy mentioned are not approved for use on pregnant women because the silver nanoparticles could actually kill like the embryo because that has like so little cells. So the silver nanoparticles won't be damaging to like an adult or in the long run because we have a lot of cells.
Did you get told about the blue person?
Oh yeah.
Oh,
Okay.
right.
So there was actually a man who consumed too much silver, and I don't really remember how he did that, but
Drank it.
Drank
he,
it.
he drank the
I'm not
silver.
positive.
Yeah, and so he actually turned blue.
Yeah.
Yeah.
But he was fine. So oddly,
Somehow.
it's apparently really not toxic to humans. Okay, one more question. Going once? Okay.
So I was wondering if you could, sorry, not close enough. I was wondering if you could explain a little more the difference between the middle picture and the picture that is audience right. It seemed like in both cases you were talking about ions going in. It's just that the third one, that was the silver powder. And so they were coming out of the clump. And it wasn't clear to me why the version coming out of the powder with the clump would cause less damage, which seems to be what the picture is showing here.
Right. So ultimately, like the ions are what's causing the damage, but like in the far right picture, they're coming off at a lot slower rate because they have to break off and then enter the ion channel, whereas the ions just have to enter the ion channel from the solution. So just because of the slower rate that they enter, that's why they cause less damage.
Yeah, there's just way less of them in
Yeah.
the end. Okay, so I will ask you, what are the limitations of this analogy?
So it didn't really depict, I guess, how the actual experiment was performed, like with the manometer and like measuring the cellular respiration. Like it does show like how the damage differs between the different types, but it doesn't show like how we measured that, which is how we actually did that in the experiment.
Any other limitations?
It doesn't show, I guess, the mechanism by which it causes the damage, like within the highly reactive, like oxygen species.
Yeah.
Like
Yeah,
within
it
the
also
cell.
doesn't really show the mechanism for going in, just different doors. So it's not trying to portray that part of it. Okay, excellent job, guys.